This is my second class on genetics. This is my website easyscience.weebly.com. You can see some more videos over here, and this is my YouTube channel link. You can see videos over there. You can keep comments, you can ask questions, and please like my channel. So now in the second class of genetics, I would like to talk about epistasis. Alright, so here we start. Epistasis. Now this epistasis, we all know about the Mendelian ratios, which is the classic ratios for the monohybrid and dihybrid cross. And for the dihybrid cross, the phenotypic ratio is 9:3:3:1, and it is a very famous classic ratio of the phenotype for the dihybrid cross. And what happens in epistasis, that we get different kind of ratios which is derived from this original ratio of the Mendelian dihybrid phenotypic ratio. Anyways, so let's start with different kind of epistasis which we got, which the scientists got due to the experiment. Mendel got this kind of ratio when he considered two different characters which was denoted by two different genes, two different alleles, but the gene was same. That means if this was the gene, suppose this was a gene and there were two alternative forms, one is A and another is small a, and this capital A was dominant over the small a, so this one was the dominant allele and this small a was the recessive allele. So in each and every character that we had discussed in the Mendelian cross and everything, all the characters were controlled by a single gene which had two alternative forms. Now during those characters, when scientists started to do different kind of experiments, they came to face different kind of problems, as they were not getting this classic Mendelian ratio, rather they were getting some different kind of ratio, suppose 15:1. That was again 16. This was the total numbers of ratios was 16, and here they also got 16, but the ratio was very different. Sometime it was 12:3:1, sometime it was 9:3:4, sometime it was 13:3:1. So there were different kind of ratios that that the scientists were getting, and with time they understood that no, there was not only a single gene involved, but some characters were controlled by more than one genes. So here came the concept of epistasis. Now let us straight away go to the epistasis, and there are different kind of epistasis that we will be discussing here. Here the first one is the dominant epistasis. Alright, here is the dominant epistasis, and the ratio that we usually get is 12:3:1. Now when this kind of epistatic interaction we can see is that when suppose there are two genes involved, one is A and one is B, and both of them have A and capital A and small b and capital B. These are the allele forms of these two genes, and definitely this capital A is dominant over the small a and the capital B is dominant over the small b. Now when the dominant allele of one gene masks the effect of the other allele of the second genes, then this kind of phenotype we can see. And suppose here this A is acting as a dominant gene over the B, so in that case the capital E is dominant over the capital B, and this capital A is also dominant over this small b. So when now here comes two terms that we would like to discuss. That gene that is actually dominating the whole situation, which is A here, it is called the epistatic gene, and on the other hand the B, which is getting suppressed or masked, it is called the hypostatic gene. Alright, so now if we go back to the discussion that we were in, then in that case this A is dominant over B, not only capital B but also over the small b. So in that case the genotype which is showing this or this will give the same phenotype, but if you consider this AB or AB, this will give these two will give a different kind of phenotype which is not same. So now if we again clear this space for further discussion of this topic, then suppose that this AA BB, and here is another genotype AABB, and this is a purple color phenotype. This individual flowers have got a purple color as a phenotype, and this capital A, small b as genotype, and on the other hand this flower has the phenotype of red and the genotype of small a and capital B, and which is a parent generation. Now when they are crossed together, they will produce this kind of genotype, and the phenotype will again be purple. Now what is happening here is that this color is actually depends, this color is produced when suppose there is a substance, a white substance is there. This is a specific kind of protein within the flower, and when a specific kind of enzyme is working on the substance, it will produce a product which has the purple color when the enzyme has been encoded from the gene A, and again this substrate will produce the red color when the enzyme that is working is from the B gene, and when no A or B are present, that means in this kind of a situation then no functional enzyme is present over here, so the substance will remain white. So now if we look at the different kind of phenotypes, there are three different kind of phenotype. One is P, one is R, and one is W. This P, this purple, is responsible for capital A. This R is responsible for capital B, and this white is responsible due to the absence of both of the enzymes. Now if we go to the Punnett square, and we can we now can work on this ratios that we had came across, which was 12:3:1, and this 12 we said was the dominant one, which is purple, this 3 was red, and this 1 was white. Now this purple must have a capital A, as this capital is dominant over capital B, capital A is dominant over small b as well. So now we have to find out where are those capital A which will produce the purple color. Here is the purple, 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 purple. Yes, now if we count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 12 purple. And look at this genotype, there is no capital A, only small b, so it will be red. This will be red, this will be red, and this will be white. Now if we look at this, there are 12 purple, 3 red, and 1 white, and this has satisfied our ratio of the dominant now what to say is the dominant epistasis. I hope it was helpful for you. Now see you in the